just wanted to go over some final points, a summary, if you will. We had an overview to start with, but I think it might be good to revisit just very briefly. So, a quick summary of the last three videos: glycolysis, link reaction and Krebs cycle, and finally oxidative phosphorylation and the electron transport chain. Right, so let's begin then. So, quick summary of glycolysis. Glycolysis, we started off with glucose, okay, and we ended up with two molecules of pyruvate. In the process, net, I'm not going to go through all the individual steps, but overall, what we did overall was generate two molecules of ATP from two molecules of ADP, as well net we reduced two NAD coenzymes and produced two NADHs. Okay, so net main products there, and most importantly there. Okay, and that was glycolysis. Then we had the link reaction. So what we could do is, we'll put the link reaction here. And it's important to know that, or to recall that this is happening in the cytoplasm, and from here onwards it's happening in the mitochondria. So we are talking about matrix. Okay, so what do we start off with? We start off with pyruvate, 2 times for each glucose, and ultimately we end up this time with 2 times acetyl coenzyme A. Okay, 2 times acetyl CoA, and in that process we had a decarboxylation. We lost a carbon dioxide to go from that 3-carbon pyruvate to the 2-carbon acetyl unit, and in that process we reduced 2 NADs to 2 NADH. Okay, so main products here, that, and most importantly, that. Okay, so what would be a good idea? Oh, I don't have a different color pen. Okay, what we'll do is just kind of circle the main store of energy, where the energy is being transferred. Where's the energy? Okay, so it's in pyruvate. Now that energy is in the acetyl CoA. Main, yeah? Yes, there's a bit there as well, and there. Okay, and of course there. Okay, so link reaction gave us acetyl CoA. Then we move the acetyl CoA, then goes into the Krebs cycle. So up here then, Krebs cycle. Again, this is in the mitochondrial matrix, and we start off with 2 acetyl CoAs, and it goes through that cycle, and our main product, and this is what I want to focus on here, our main product is reduced NAD and reduced FAD. Okay? Yeah. So, as we said, for each acetyl unit, we got 3 NAD reduced and 1 FAD reduced. But so 2 acetyl units, turning the cycle twice, would give us 6 NADH and 2 FADH. At the same time, we will be producing two molecules of carbon dioxide per cycle. So we've got 4 for every 2 cycles. 4C2. And remember, we also had a substrate level phosphorylation to produce ATP here, and there was 1 molecule of ATP per cycle, so we make 2 ATP for the cycle turning twice. Okay, so that's the summary of the Krebs cycle, and now the main store of the energy that we need to focus on is in these reduced coenzymes, and then they will go to the electron transport chain. Electron transport chain, and that's in the mitochondrial inner membrane. That's where that's happening. Okay, and that's not really a metabolic process per se. It's using the flow of electrons to pump protons, and because of that we get ATP produced through the ATP synthase. So how do we represent that? Yeah. Do we need to draw it out? Anyway, let's just call the electron transport chain, and I guess ATP synthase, which is not technically part of that electron transport chain. It's a separate protein. It kind of uses the main product of that electron transport chain, which is the proton gradient. Okay, so the electron transport chain produces the proton gradient. The ATP synthase takes advantage of that. As the protons move through, back down that gradient, the energy is used by ATP synthase to synthesize ATP. Okay, so we have NAD. Well, let's just say that we get electrons going in. Okay, and the electrons going in are thanks to or provided by NADH and FAD. Okay, so electrons go in. Electrons go in, but they have to come out, right? And actually, it's electrons and protons that are provided to this, so we should say H plus. Okay, so electrons go into that, H plus goes into that. You know, I'm grossly simplifying here, but you know, the protons don't really go into the electron transport chain. Anyway, electrons go in, protons go in, and importantly, oxygen is there to accept the electrons at the end of the electron transport chain, and that's a critical step. Because if oxygen doesn't accept those electrons, that chain gets kind of blocked up with electrons and doesn't function. So what comes out is water, but oxygen is combining this. So oxygen, let's just call that O2, plus the electrons and protons gives us water. Okay, so this is the summary then of aerobic respiration. These things will happen like this as long as we have oxygen. Actually, for each oxygen molecule, we'll have 2 water molecules, and I've just made a correction there. For 2 pyruvates that we convert into 2 acetyl units, we'll lose 2 carbon dioxide molecules. Okay, so I think the stoichiometry is okay here. All right, it's going to get a bit shady regarding the protons and the electrons, but you know, here we don't need to know about numbers. Here we just need to know about how the process works, essentially. Okay, and so So as I said, we can underestimate the role of oxygen in this process, because I think we did go through it a little bit in the previous video, that if oxygen is not there to accept these electrons, then the electron transport chain will stop. If the electron transport chain stops, we don't get ATP production. So actually, what we should have is ATP being produced here. And the other very important thing we have is the regeneration of FAD and NAD that have been reoxidized. Okay? Because it's only these that will then be able to provide, maybe, I don't want to overcomplicate this, but it's really important that we appreciate that. It's the reoxidation of NAD and FAD at the electron transport chain that then provides the oxidized NAD and FAD that is required for the Krebs cycle. So this NAD and FAD reduced is only possible because we have oxidized NAD coming in, available to accept those electrons and protons. Okay? And that is true even at earlier stages like the link reaction. Okay? And to a certain extent here. Okay? Right, so therefore, because oxygen is therefore allowing electron transport chain, Krebs cycle, and link reaction to occur in the matrix. Remember, if that stops, we don't have NAD for the Krebs cycle, we don't have NAD for the link reaction. Okay? So we don't have any coenzyme to be able to reduce. And if we can't reduce that coenzyme, that reaction can't happen. If we can't reduce coenzyme here, Krebs cycle can't happen. Okay? So link reaction, Krebs cycle, electron transport chain dependent heavily on the presence of oxygen. This is really critical, okay? And it kind of allows us to talk about the last little snippet that I haven't covered yet, which is anaerobic respiration. Because there are many situations where the supply of oxygen is not really sufficient for the respiratory process. So the rate at which we need ATP, oxygen isn't provided at that rate. Okay? So we need to have a way to still make ATP without oxygen. Okay? So that's what we're going to look at next. How do we still make ATP without when oxygen is not present? Because for things like, for intense activities, the ATP that you use up is much greater than what you're producing by aerobic respiration. Okay? So when your oxygen isn't being supplied at the rate that it should be to the cells, or that it needs to be for aerobic respiration, you still need a way of generating ATP. Okay? And we'll look at how the cell manages to do this and the significance of that process next. Okay? So just remember, for the next part, what you need to appreciate is that in the absence of oxygen, these three stages cannot happen. Link reaction, Krebs cycle, electron transport chain. So how does this carry on, and what are the kind of other considerations that we need to take into account? Okay, so that's the next bit. Okay, so I pondered getting rid of this to look at the anaerobic respiration, but I think it might be more useful, impressive, to just kind of add it on here, okay? Because then it will be in context. So it's like, this is the normal route of what happens, but then what we discuss here is going to be what will happen when oxygen isn't present and these things can't happen. Okay, so let's just draw a red line here. Okay? Because what we're saying is that to a certain extent, glycolysis can still occur without oxygen. But there is one problem there. And the problem is having a supply of reoxidized NAD coenzyme. If we look at the glycolytic pathway here, in summary, that glucose can be converted to pyruvate, and we do get ATP produced. And it's not directly dependent on anything that is dependent on the oxygen being present. However, there is one problem here. So let's just say that oxygen supply stops. Glycolysis can still occur as long as we have a supply of glucose. Problem is that it can only carry on, and we can only make a small amount of ATP in comparison, but the problem is that for the glycolysis to proceed to pyruvate, you still need a supply of reoxidized NAD. Okay? Now, this pathway produced 2 molecules of reduced NAD, and that NAD contributed to the electron transport chain at some point. Okay? And in that process, it got reoxidized back to NAD, and therefore we replenished our supply of NAD that could take part in the glycolytic process, that could provide us with pyruvate, but importantly, ATP production. However, in the absence of oxygen, we'll keep making pyruvate, we'll keep making enough ATP, fine. But as soon as we run out of reoxidized NAD, even glycolysis will stop. Even glycolysis will stop. So we need to have a way to regenerate this NAD without oxygen. And that's essentially what the anaerobic kind of destinations of pyruvate are really for. It's simply a way to use up this NADH, remake NAD so that glycolysis can carry on. Okay? Remember, the only way for us, when oxygen is present, the way we regenerate our NAD is by the NADs, the reduced NADs delivering their electrons, their protons into the electron transport chain oxidative phosphorylation stage. But if in the absence of oxygen, if that isn't happening, how are we making this? And if we're not making that, how can this carry on? And if this can't carry on, how are we generating ATP? We have then no way of generating ATP, and we will die. Okay? Cell functions will stop, and therefore tissues don't work, therefore organs don't work, therefore organ systems don't work. You're dead. Okay? So we need to have a way of regenerating NAD, and this is what the 2 processes of anaerobic or fermentation really are for. Therefore converting pyruvate to something else, because obviously it's not going into Krebs cycle anymore, or link reaction Krebs cycle anymore. So what do we do with the pyruvate? It's really to regenerate this NAD. So let's see how that happens. And I'm going to be using a red pen to indicate that this is happening in the absence of oxygen. Okay, so let's see what happens to pyruvate in 2 different types of organism. Pyruvate. So the first one is in animals, in our bodies, and it's called lactate fermentation. So in lactate fermentation, pyruvate is converted to lactic acid or lactate, however you want to call it. But in that process, we use up the NAD. Okay, so the NADH is used up. So the hydrogens from the NAD are used up in this conversion. And so we've regenerated reoxidized NAD, and that NAD can go back to the earlier stages of glycolysis and take part in the dehydrogenation steps required to keep this process going. So as long as we keep that process going, we're making ATP, and as long as we got enough ATP going, even though it's comparatively smaller, it's sufficient for short bursts of kind of intense activities. Okay. Right. So we've got lactic acid, and the enzyme that's carrying this process out is called lactate dehydrogenase. Lactate dehydrogenase, sorry, I can't read that. Lactate dehydrogenase, okay? So, that's one way to regenerate reoxidized NAD. In yeast, the process is slightly different. In that case, what happens to the pyruvate is called alcohol fermentation. This has two steps. Pyruvate is first decarboxylated to a compound called ethanol. Okay, so it's decarboxylated, CO2 there. That step is carried out by pyruvate decarboxylase. Pyruvate decarboxylase. And the ethanol is then hydrogenated to ethanol, okay? And in this step, importantly, this is the main one. In this step, NADH loses its hydrogens, and we regenerate NAD. Okay, so this process is catalyzed by the enzyme ethanol dehydrogenase. Ethanol dehydrogenase. Okay, so but importantly, in yeast, in this process, is again we've produced ethanol, and that's the kind of source of the alcohol in yeast fermentation. But importantly, the main thing is that we've regenerated reoxidized NAD. This reoxidized NAD can go back into the glycolytic process and keep that process going, so that even though we're generating small amounts, we're still making ATP, even when oxygen is not there. Okay? So that's the whole point. That is the whole point. Okay? That's it, guys. And any questions, do ask me. But I'll say thank you for watching and good luck.